Hello, my name is Bojan Papić, and I come from the Institute of Microbiology and Parasitology at the Veterinary Faculty, University of Ljubljana, Slovenia. So I would briefly like to present to you our work on whole genome sequencing of Paenibacillus larvae. Paenibacillus larvae is a gram-positive bacterium, and it is the causative agent of a very serious disease of honeybees. On these figures, you can see some characteristic clinical symptoms of the disease. And the most characteristic is the development of a ropey mass inside the honeycomb cells. So whole genome sequencing has really revolutionized the field of microbial typing, including bacteria. And there are two general approaches to microbial typing. One is an allele-based approach, in which a unit is a locus or a gene. And the second one is a SNP-based approach, which is in which the unit is a gene, a nucleotide or SNP. And because core genome and whole genome multilocus sequence typing are more appropriate for standardization, we decided to construct a stable scheme for Paenibacillus larvae. And we also showed that both approaches are generally interchangeable. So to assess the intra and inter-outbreak diversity of Paenibacillus larvae, so within and between outbreak diversity, we typed 59 isolates. And of these, 40 originated from a single beekeeping practice, which included 3 apiaries. And these are the results of 2 different clustering algorithms. On the left, you can see the wgMA clustering or tree, and on the right, you can see the minimum spanning tree. And basically, both clustering algorithms showed or revealed 2 clusters. One is denoted in red, and the other one is denoted in blue. And we observed that the maximum diversity within a single beekeeping practice reached 11 allele differences. The maximum diversity within an outbreak cluster reached 34 allele differences. And the minimum difference between the outbreak clusters was 51 allele differences. And these isolates are the epidemiologically unrelated isolates that clustered between these 2 clusters. So in summary, we observed that by applying the threshold of 34 allele differences, we can accurately delineate the outbreak clusters. However, these links should be always confirmed by the underlying epidemiological data, and adding the epidemiologically unrelated isolates into the analysis may help us in outbreak cluster delineation. And also, the diversity within a single beekeeping practice should always be taken into account. What are the current gaps in knowledge? And I was hoping to get your feedback also on this topic during this session, is that we do not know the evolutionary rate of Paenibacillus larvae. There is a lack of sequenced archaic isolates, also lack of evolve and resequence studies, and we do not know what effect does the spore formation have on the evolutionary rate, because this microorganism is a spore-forming microorganism. So with this, I would like to conclude my talk, and thank you for your attention.